Enteric fever, potentially fatal multi-system illness caused by Salmonella typhi, typhoid fever, and Salmonella paratyphi ABC causes paratyphoid fever. Classification and nomenclature, which include three classification: clinical, antigenic, and third one is molecular. Now we read about the clinical classification, which is also called the oldest classification. Divide Salmonella into two groups: typhoidal Salmonella, including serotypes S. typhi and S. paratyphi, restricted to human host, cause enteric fever, and non-typhoidal Salmonella, including remaining serotypes, colonize intestine of broad range of animals, also infect humans, causing gastroenteritis and septicemia. Antigenic, based on different somatic O and flagellar H antigen, serogroups based on OA antigen, Salmonella classified into serogroups. Serotypes. Each serogroup is further differentiated into serotype based on type of flagellar antigen present. Molecular. Based on DNA hybridization study, two species S. enterica, six subspecies, most of pathogenic typhoidal and non-typhoidal Salmonella. Serotypes are placed under species, enterica and subspecies, and second one is Salmonella bongori. Now about typhoidal Salmonella. S. typhi and S. paratyphi ABC, which cause enteric fever. Pathogenesis transmitted by oral route through ingestion of contaminated food or water. These factors include conditions that decrease stomach acidity, less than 1 year age, antacid digestion, prior Helicobacter pylori infection, intestinal integrity, IBD prior GIT surgery. Now the mechanism of entry through epithelial cells. Salmonella can trigger formation of membrane ruffles on cell membrane of M cells. Now these ruffles reach out and enclose the adherent bacteria within large vesicle. This process of uptake is called bacteria-mediated endocytosis. This bacilli remains inside vacuoles in cytoplasm, and then Salmonella containing vacuums cross epithelium, reach some mucosa, phagocytosis by macrophages. Now this Salmonella typhi induce alteration on its surface, so it no longer susceptible by macrophages. Salmonella inside the macrophages and then spread via lymphatics, inter bloodstream, and then spread through reticuloendothelial tissue and other organ where further multiplication takes place. Now this secondary bacteremia leads to onset of the clinical disease. Clinical manifestation of enteric fever, largely extraintestinal, incubation period 10 to 14 days, fever rises gradually to higher level with every spike, then fall down but not normal. Headache, chill, cough, spitting, myalgia, rashes, rose spots, fan, salmon colored. Early manifestation include abdominal pain, nausea, vomiting, constipation or diarrhea. Important site: hepatitis, splenomegaly, epistaxis, and relative bradycardia. Neurological manifestation: meningitis and cerebral ataxia. Epidemiology: humans only, and transmission by ingestion of contaminated food and water. Factors: poor sanitation, contaminated water, food and drink, lack of hand washing, and poor toilet access. S. type infections more than S. paratype infection, and carriers are of two types: fecal and urinary. Now about lab diagnosis. Specimen collection: first, blood or bone marrow culture, first week of illness, and the serum for serology used in second or third week of illness, and the stool and urine culture used in third or fourth week of illness. Culture and identification. Blood or bone marrow culture. Ideal method for diagnosis: in the first week of fever, which become positive in about 90% of cases. Blood culture bottle. 8 to 10 milliliter of blood may be collected in blood culture bottle, either conventional bottle or automated bottle. Incubation: blood culture bottle are incubated at 37 degrees centigrade. Salmonella or non-fastidious growth occur within 24 hours. Colony appearance: in blood agar, non-hemolytic most colonies, and in Mekong agar colonies are round, translucent, pale, and non-lactose fermenting. Now about stool and urine culture. थर्ड और फोर्थ वीक ऑफ इलनेस में यूज़ किया जाता है. इवन बिकम पॉजिटिव इफ पेशेंट इज ऑन एंटीबायोटिक्स ट्रीटमेंट. फॉर डिटेक्शन ऑफ करियर्स यूज मेकॉन की अगार फॉर यूरिन कल्चर एंड फॉर स्टूल कल्चर. Enrichment broth like gram negative broth and selenite F broth, and the selective media like Mekong ki agar, etc. Culture smear and motility testing. Gram stain smear, gram negative, non-sporing and non-capsulated, and the bacilli identification by automated identification systems such as Multitop or VTEC, or by conventional biochemical test catalase positive and oxidase negative, and Dole test negative, and Satres test also negative. Detection of serum antibody by Vidal test in 2 to 3 weeks of illness. Tube agglutination test is used. Detects antibody in patient serum against antigen of S. typhi and Salmonella paratyphi. 4 antigen is used: O and H antigen of typhi and H antigen of para A and para B typhi. If person have a salmonella typhi, then TU and TH antibody increase, and if S paratyphi A, then TU and H antibody increase, and if S paratyphi B, then TU and BH antibody increase. Antigen detection method, biology and molecular method, nested PCR, differentiate method.